This is the microbe identification project from General Genetics Lab, made by Michaela Moran and Raphael Milangi. In this experiment, an unknown sample of bacteria provided by the instructor and an environmental sample of bacteria were cultured on TSA plates and had their DNA extracted for sequencing to determine their identity. In this lab, we basically had three phases. We had phase of extracting DNA by using several reagents and enzymes to separate the cellular components from the genetic material. Next, we had PCR, which is the amplification of a specific target gene by using specific primers, polymerase, buffers, DNTPS, and different temperatures to denature, anneal, and extend the gene of interest. And finally, we had the step of sequencing, where we took the target gene sequence, the unknown ones, and compared them to a bunch of known sequences on a database such as BLAST. We also had a step of gel electrophoresis to visually observe if the PCR was successful in amplifying said target gene. The gene of interest is the 16S rRNA gene. It's about 1,500 base pairs long, and in the PCR step you'll need specific forward and reverse primers for the amplification of this gene. The 16S rRNA gene does code for the small subunit of the ribosome, which is involved in the protein-making process, specifically translation, and it's conserved throughout all the species of bacteria. Since it's conserved throughout all the species of bacteria, if we have an unknown sequence of the 16S rRNA gene, it could be compared to known sequences of bacteria that are featured in a database such as BLAST, and by viewing all these comparisons, we could figure out the identity of the unknown bacteria. More information on PCR. PCR requires the usage of buffers, DNTPS, which are free of nucleotides, and as well as the forward and reverse primers. We had two sets of primers. We had a long primer mode where we are using a 27 forward and 1492 reverse primer. This would have given us a 16S gene about 1,400 base pairs long. Then we also had the short primer group of 200 forward and 518 reverse. This would have given us a 16S gene of about, give or take, 300 base pairs. We also used the Taq polymerase, and this enzyme will add new complementary bases to the fragments of the DNA during PCR, specifically when the primers have attached during annihilation where the temperature is cooled, then the polymerase will be able to add new complementary bases to the template strands. We use the Taq polymerase because it's from a bacterial species, Thermus aquaticus, which is able to live in harsh temperature environments. This allows for the enzyme to not be as susceptible to the high temperatures needed for that first step in PCR, which is denaturing the DNA or separating it using high temperature. A negative control was used in the experiment in both the AdvoBead and MasterMix runs, and the negative contains the same DNTPS buffers, the Taq polymerase, the forward and reverse primers, but it's different from the actual PCR solutions in which it does not contain the template strands or the DNA extracted from the bacteria. And if the negative control is used, it may reveal false positives in the solutions with the DNA, possibly to a master mix contamination, or if one of the components itself, for example, the buffers might be contaminated with genetic material, or the DNTPS or the forward and reverse primers, they may have gotten contaminated when the solutions were being mixed together. that could be determined if the banding in gel electrophoresis does appear in the negative control well. More steps, again, the culturing of the bacteria and the environmental swab that we did. It was off of a cafeteria kiosk. It was unsuccessful in culturing any bacteria, so our group had to borrow from another group in lab for an environmental swab. And we had to do PCR actually in 2 rounds before we had any feasible results, but in both times it was a failure, so we also had to borrow from another group's gel electrophoresis that did have some banding on it for the rest of the study. The DNA extraction, which was done before PCR, ran successfully, and we didn't have any errors in that sense. But the original slant A sample did get lost in storage, and so the lab instructor did provide us with a new slant K, which was used since losing the slant A in between the extraction and the PCR. And of course we had the sequencing at the very end where we were given 4 unknown DNA sequence ABI files. Since our gel electrophoresis and our PCR runs, both of them were unsuccessful, so we were given 4 unknown sequences from the instructor, and we just compared those against those in the BLAST database. The results, these are the absorbances and the DNA purity ratios for the samples used in PCR. Slant K, this is going to be like the unknown bacteria sample. The DNA extracted had about a purity ratio of about 1.6, and the absorbances in ng over microliter, we had 84.8 on the first drop and 113 on the second drop of slant K. The environmental samples purity ratios were around 1.97 and 2, while its absorbances were 42.1 and 41.1 for drop 1 and drop 2 of that environmental samples DNA. The original slant A TSA plate after incubation alongside our original environmental swab could be seen on the left side of this slide. As you can see, the TSA plate on the top is the slant A, where we did have successful culturing of bacteria that we could extract from. However, the environmental swab, which was taken from this location in the middle in that kiosk in the cafeteria, it did not have any growth, so we're going to borrow from the other lab group. And in the right picture, we have the location on their petri dish with some colonies where we got ours for the rest of the test. These are the results of the DNA extraction on the left, and then our PCR products for the first round. As we have a negative control in the far left of the PCR picture, then we have the environmental PCR mixture, then we have the unknown bacteria mixture on the far right of that PCR section. And for the first round, we were using the long primer. So this is the 27 forward and 1498 reverse. The original PCR run did fail to display an expected presence of banding in the wells that contain the environmental and even the unknown banding, the unknown PCR. The banding for a fragment of the 16S-rRNA gene that would have been around 1,400 base pairs long was not present, even though it was expected to be there if PCR was successful. Since it wasn't present in the gel, it would have been near around the top, since the larger fragments of DNA wouldn't travel to the positive end in the electric field as easily as a smaller fragment. We did not see a band at the top for wells 3 or 4. Those were the wells that had the bacterial PCR solutions in. We count from left to right. And we did actually get the expected no banding in the negative control, which wouldn't have had any DNA template inside of it. So we did not get banding there for the gel electrophoresis, which was the expected, and that means that none of the reagents that were added in the PCR mix were contaminated with the genetic material. After a redo of the PCR with both EDVO beads and a master mix, the scans of the gel electrophoresis had the same similar failed result as in the picture right there, and these displayed the similar lack of the 16S rRNA gene being successfully amplified. This is a gel electrophoresis scan borrowed from another group in lab, and they did have banding. It was expected at the wells from the left to right. We had in wells 2 and 3, we had the log primers with bacterial. So this is the unknown slant of the group, and then the environmental is in well 3, and these were with the long 27F1498 reverse primers. The bacterial did not have the expected 1471 base pair fragment of the 16S rRNA, but we did get one in well 3, which was the environmental. We had a banding of 1471 base pairs, which was expected because we're using the long primers. And we had the expected negative appearance of a band in the negative control because there wasn't any DNA contamination in any of the reagents for them. In the short primer side, we are expecting a band much closer to the positive end of the electric field on the gel, since the short forward and reverse primers would amplify a shorter region of the 16S rRNA gene. And here we have in bacterial, which is well 5, we have a 318 base pair band present, which was expected. And we also have that same thing in the environmental. It was expected. We still have that 300 or so base pair banding. But in the negative, we have an unexpected 318 base pair band as well. Since the negative isn't supposed to contain any of the DNA template or even the amplified gene, since we didn't add any of it into a negative control for the PCR, this observation of the unexpected banding there points to a possible false negative, or it could also point towards a possible contamination of the reagents used in the PCR mixture, to specifically the primers, the forward and reverse primers in the EDVO bead samples. And that primer may have been contaminated with some genetic material, and therefore a band showed up in the gel that was borrowed. After the sequences of 4 unknown bacteria were given to us by the lab instructor, they were cleaned up in computer software and then compared towards the BLAST database for known genetic sequences of other bacteria for their 16S rRNA gene. From that data and the comparison between the species and the unknowns, a phylogenetic tree is possibly built. And from the data of the compared 16S rRNA gene sequences provided by Nucleotide BLAST from NCBI, we had the 4 following species. We had 2 bacillus species, Weedmanni and Megatherium. These are both the closely related species to each other, since they're from the same family, Bacilliaceae. And the second most closest species to the bacillus, the 2 bacillus species, it would be the Staphylococcus aureus bacteria, since it is from the same order as the bacillus species, but it's just only from a different family, genus, and species. The outgroup of the bacteria that were compared is the Proteus harsuri, since it's classified in a different phylum than the other 3 species. As you could see, Staphylococcus and the 2 bacillus species are from the phylum Bacilliota, and Proteus harsuri is from the phylum Pseudomonadata, and it's a separate branch in the phylogenetic tree. It's separated from the other three species, and therefore it's the outgroup. It's the least related to the other species. More information on the bacteria and the sequence number that they most compared to. Sequence number 9 was most related to the Staphylococcus aureus sequence for this 16S gene. And Staphylococcus aureus is a gram-positive bacteria. Its habitat is the respiratory tract, and it's actually a main component of the microbiota, which is the natural biome of microbes on the human skin. And the significance of this bacteria is that it can become a pathogen if it enters the skin through an infection, or if the skin gets ruptured and it goes into a layer of the skin where it's not supposed to. This becomes a pathogen. In the unknown sequence 10, BLAST database found it most comparable to Proteus hasuri, also known as Proteus vulgaris. Now this is a gram-negative bacteria, and it's found in the intestinal tracts of animals. The significance of this species, it's able to produce sulfides, and it could cause urinary tract diseases. Its ability to produce sulfides stems from it being a player in the sulfur cycle. This is how sulfur is transformed throughout an ecosystem or an habitat as it goes from different organisms and different locations in that said ecosystem. Number 11 sequence was most like the sequence of Bacillus weidmanni in the BLAST database. Bacillus weidmanni is a gram-positive bacteria. It's found in raw milk, and it's known as a cytotoxic species. Sequence number 12 was most related to Bacillus megaterium, according to the BLAST database. This is a gram-positive bacteria, and it's found in a wide range of aerobic areas, such as wetlands, seawater, ponds, and even in the soil. And it has the potential to participate in the nitrogen cycle. This is similar to the sulfur cycle, but it's just the recycling and the movement of nitrogen in the environment from species to different forms throughout the ecosystem. From the observations and the carrying out of the lab, it can be concluded that if bacterial samples are cultured, their DNA could be extracted. If a conserved gene such as the 16S rRNA is successfully amplified through PCR, the identities of unknown bacteria could be found if the sequences are compared within the BLAST database of known genetic sequences. The more similar sequences of unknown bacteria are to a specific bacteria that has a known sequence in the database, this could support the hypothesis that the unknown species is the most likely or most related species given by the BLAST database. Specific primers must also be used in order to isolate a specific gene within a template material, such as DNA for PCR. If this lab were to be repeated, a different environmental sample could have been cultured and DNA extracted for the sequencing of its 16S RNA could have been sought, and PCR could have been performed on this gene and amplified, and the sequencing could have been done for the actual gene instead of just 4 sequences given out by the lab instructor. And fortunately, there was also a time constraint throughout the whole lab, where PCR could have maybe have been redone one more time, but due to the time constraints, it was unable to do so. Thank you so much.